Okay, welcome everyone. Today we're going to be talking about adaptations of skeletal muscle to aerobic and anaerobic training. This is my good friend Ben Pekulski getting ready to compete in the Arnold Classic, and we're hoping he wins this year. So let's talk about adaptation. Here you have an individual who could bench press 315 for 6 reps. They train and they get a strength training adaptation to where they can actually lift 315 for 8 reps. So what do you think is causing that adaptation? Well, of course, one thing you know is that it could be what? Central, right? Neural. However, it also could very well be that this individual hypertrophied or increased in size. Now, is all hypertrophy the same? This is sort of more of a controversial topic, but muscle hypertrophy refers to essentially an enlargement of the muscle from an increase in the cross-sectional area of the existing fibers. So here we have Phil Feister, a powerlifter / strongman, who likely trains completely different than Sean Roden, who won or actually took third in the Mr. Olympia contest as one of the favorites to win the Arnold Classic this year. So let's talk about how muscles grow. Look at this picture. This is essentially a longitudinal picture of a muscle fiber. And what do you notice? What's unique about this muscle fiber? Well, what you see is that it has several nuclei. Okay? What happens is that, why does it have several nuclei? Think about that for a second. Well, if you circle that nuclei controls the transcription or the development of instructions for proteins in a limited region of the muscle. Okay? So that region ranges from around 1,400 to 2,000 micrometers. It's about 1,400 to 2,000 micrometers. So if we look, the muscle is clearly a lot larger, and that area I'm circling there is probably around 2,000 micrometers. Or actually no, more about 1,600, let's say, for the purposes of this. And if we go here, we blow that up, sort of in diagrammatical format there. If you lift weights, what's going to happen? You're going to increase protein synthesis, and that's going to build new muscle. However, at some point in time, if that hit that ceiling of 2,000 micrometers, you couldn't grow any further with the current nuclei you had in your muscle. So what do you have to do? What you have to do is you have to add more nuclei. So basically that's the key. You have to add more nuclei to continue to grow. All right, so why do I have this random picture in there of the Earth and the Moon? Well, the Moon is essentially the Earth's natural satellite because it's on the periphery of the Earth. Well, if you look here, this is some data that we collected back when I was in grad school. We're using a light microscope, and you can see the individual muscle fibers right there. Okay, if you look at the arrows, if you look at the outside of the muscle fibers on the very periphery of them, you see these small cells that are dominated by a large nuclei. Since they're on the periphery of the muscle, just like the Moon's on the periphery of the Earth, we call them what type of cells? Satellite cells. And satellite cells' role is to donate their nuclei to muscle. So here we have a resting muscle fiber. We say it's quiescent or resting. But if you induce trauma to that muscle, whether that's from injury, burn, or exercise, then those satellite cells that are right there will begin to divide. And they'll divide or proliferate. Chemotaxis means that they actually migrate to the injured area of the muscle. They will fuse to the muscle and donate their size and their nuclei to the muscle fiber. So basically, satellite cells, in response to trauma, divide and donate their nuclei to muscle so you can support further growth. So now muscle growth is a factor of cell signaling. So if you look here, we have the nucleus, which essentially controls the transcription. Blood-borne growth factors and intramuscular growth factors will ultimately help transcribe new proteins to grow. So again, muscle growth can occur by increasing protein synthesis, which makes the cell bigger. An increase in nuclei by activating satellite cells is also needed to support a larger muscle. Increases in circulating and primarily actually intramuscular growth factors, as well as nutritional interventions, ultimately trigger these processes. So think about it. How do bodybuilders train? We've talked about this, and how do powerlifters train? Well, bodybuilders in general train, their repetitions are moderately high, like 8 to 12 reps. They have shorter rest period lengths, like 30 to 60 seconds. The energy system they stress is glycolysis, and their volume is generally high. Whereas the powerlifters lift low repetitions, they rest a very long time, like 5 minutes, some rest 10. Their main energy source is the ATP-PC system, and their volume is low. So, there are two general types of hypertrophy that can occur, and this is more of a controversial topic, but I don't see how it's controversial at all, because essentially a muscle can grow by adding myofibrils, this is a contractile element of the muscle, or it can grow if it's stressed energetically by adding mitochondria, or a number of enzymes, or even glycogen, right? So mitochondria itself has mass, and that mass can increase the size of the cell, right? So what we have pre-training cross-sectional area all the way to your left. Okay, if you lift primarily heavy, then what you're going to end up doing is primarily increasing the myofibrils. If you lift more of a higher repetition range, or like bodybuilders, you may increase the sarcoplasmic, what's called sarcoplasmic hypertrophy, which, although controversial, really is referring to increases in mitochondria. Okay, so what will happen is that a powerlifter will primarily increase the myofibrils, and a bodybuilder needs to be stronger, but they need to sustain contraction, and the sustenance of contraction will be driven primarily by increases in things like mitochondria, while the ability to increase force is going to be increased by myofibrils. So bodybuilders are both, powerlifters are both, but it's really to a major extent myofibril. Now, this is a little bit tricky graph here, but basically what it's showing is that if you look at type 2 muscle fibers, they're hypertrophying a lot more, around, say, 30% compared to like 20% with slow twitch muscle fibers. So there's quite a large difference. So the point is that type 2 fibers have a greater capacity to grow than type 1 fibers. So if you look here, if you look at weightlifters and powerlifters, but they train fairly similar in that they're lifting at a heavy repetition range. And if you look at the majority of their muscle size, and you have type 1 and type 2 fibers, you see that the size of the muscle is primarily driven by type 2 fiber changes. That's because they're not lifting very high repetition range, so they're likely not going to stress the type 1 fibers and cause them to hypertrophy. On the other hand, bodybuilders have dramatic increases in type 1 hypertrophy, but they're clearly not maxing out type 2 hypertrophy. So this is why, but bodybuilders will train that 8 to 12 repetition range. So to bring up the type 2s, what do they need to do? They need to throw in some heavy lifting like powerlifting. So for the majority of the time, maybe 50 to 80%, they might be training hypertrophy style training, but they need to have at least 20-30% where they're lifting heavy as well to maximize the type 2 fiber hypertrophy. Now this is a very difficult graph to understand. What it's showing is that when you train from 40 to 95% of your 1RM, what happens is if you look at the thick dotted line above, what you're looking at is essentially type 2A muscle fibers. Okay, if you look at the dark line, That's type 2, really type 2X fibers. What it's saying is that at any given intensity, type 2X fibers decrease in their percentage, and type 2A fibers increase in their percentage. Okay, well, this is traditional lifting. What that suggests is that there's an interconversion in which you shift from type 2X to 2A. And you want to think about 2X as basically sort of being like the out of shape type 2 fiber phenotype. Yes, so initially they're larger, but they're harder to recruit, so they're out of shape. Once you recruit them, they take on the enzymatic properties of type 2A. And so most powerlifters, surprisingly, don't have a lot of 2X fibers. They're pretty much all 2A. If you notice with traditional lifting, you don't get much of a change in the type 1 fibers. Those stay the same. Now again, this is your textbook answer. So your textbook answer is this. With traditional resistance training, okay, type 2X, I know it shows 2B, but it's really 2X, converts to 2A. Whereas type 1, or the slow twitch fibers, stay the same in traditional. However, Liao in 2003 showed that you could actually interconvert the type slow fibers to the fast twitch fibers. So we had one traditional group where they lifted heavy, so they did 5 sets of 3 RM lifting. So that's the graph over to the left, the heavy group. And they lifted this way for several weeks. What I want you to notice is that the 2A fibers go up and the 2X go down. What does this suggest? This suggests interconversion, that 2X is turning 2A. He had a second group that was a combination group. On day 1, they lifted 5 by 3 RM. On day 2, they did 10 bench press throws at 30% of their 1 RM. And day 3, they did 3 sets of 10 clap push-ups. Okay? So those are what's called ballistic or power training. The bench press throw is a ballistic movement. The clap push-ups is a ballistic movement. It's explosive. It's done with lighter weight. They also had heavy day. So the power type of movements requires fast twitch fibers most definitely. If you look at the combination group, the slow twitch fibers went down and the 2A fibers went up, but the 2X stayed the same. This suggests what? That when you train for power, that you have an interconversion between slow and fast fibers. Now I want to emphasize something to you though. If you were to do endurance exercise, long duration endurance exercise, what would you think would happen to this adaptation? Well, it would be stopped essentially. You wouldn't get this adaptation. Okay, so the key is this could be very important to sprinters, and it demonstrates they need to train explosively. So McDougall did a study where he was looking at mitochondrial density and primarily power lifters, heavy resistance training, and basically mitochondrial density is how much mitochondria you have per unit area. What you see McDougall finds is that after 6 months of purely lifting heavy and not doing any cardio, that resulted in a 33% increase in muscle fiber size, but there's a 25% decline in mitochondrial density. What that means is that the myofibril growth was outpacing the mitochondrial growth. And again, think of mitochondrial growth as that's what some people call sarcoplasmic. While that name's controversial, we do know mitochondria increase. And in this case, when you lift super heavy, they're not increasing to the same extent as the myofibrils. So your density goes down. That's why if you're a purely power lifter, you're lifting 1 to 5 reps, you do no cardio, you avoid any, taking the stairs, things like that, you're going to feel out of shape. You're going to be breathing and huffing and puffing. So whereas the bodybuilder or the person who combines concurrently resistance training and endurance won't. Something similar happens with capillaries. If you look at capillary density, you look at non-athletes, that's capillary density, is capillaries per area of muscle. Powerlifters go down because their training doesn't induce capillarization to a high enough extent to keep pace with the myofibrils. Bodybuilders stay the same. Now, does that mean that bodybuilders aren't increasing capillaries or mitochondria? No. They're increasing it a ton because they have double the muscle that the non-athletes have, as far as cross-sectional area. And then the endurance athletes are not increasing the myofibrils that much. They may be going down, but their capillaries are going up, so their capillary density is higher than all the other groups. Now let's talk about muscle damage. What is muscle damage? Well, muscle damage occurs when there's a disruption of the integrity of the muscle cell, leading to a decrement in muscle performance. So here we have some damaged muscle tissue. To the left, we have normal muscle tissue. But then after you do an exercise bout, look what happens to it. And what that's called there to your right is called Z-line streaming. So before the Z-lines are perfectly aligned, and then they start to strain. And you see disarray. In fact, if you look at some of the fibers there, it looks like they're absolutely destroyed. In fact, look at this right here. What's happening there? You know, what happens is we're saying we think this sarcomere is destroyed or being remodeled. And we call that a pop sarcomere. Okay? So, one of the things we look at, this is in my older lab, and basically what we did was we used this biodex here and we created a lot of muscle damage. When we do a muscle damage protocol here, if we look at the muscle, if the muscle is damaged, muscle inside has a lot of enzymes. Like creatine kinase, for example, catalyzes the reaction. That takes essentially creatine phosphate and donates a phosphate group over to adenosine diphosphate to reform adenosine triphosphate. So it's high in the muscle. If you damage the muscle becomes more permeable, becomes leaky, and CK leaks out into the blood. So the more CK, the greater the damage. This is some research that we did, and it's characteristic of a lot of studies, where you're looking essentially at creatine kinase over to your right, and to the left you're looking at lactate dehydrogenase. So that's another enzyme in the muscle. Notice that lactate dehydrogenase in all three of these experimental groups is peaking from 48 to 72 hours. And if you look at creatine kinase, it peaks at around 48 hours. So we say essentially that this muscle damage response is peaking in general anywhere from 24 to 72 hours. But we see this in the serum enzyme levels. As another study we did, where again, you're looking at soreness as a measure of damage, and muscle soreness peaks at around 48 hours. Now, this is another simple study we did. We were looking at a concept known as a repeated bout effect. What we basically did was, you're looking at muscle soreness, and you see muscle soreness goes up to 48 hours, comes back down after a first initial bout, okay? So this is the first bout. This is an arm flexor bout. We're doing incline curls with untrained people so they get sore. You bring it back 7 days later, do the same bout, nothing happens. Right? So basically we call this a repeated bout effect. Something happens in the muscle that allows it to be protected against further damage. One of the things we think is related to what's called a popping sarcomere hypothesis. So what happens is that if you're lifting, remember you have sarcomeres, and these are in series, and this is a myofibril. The weak sarcomeres, when you do an eccentric contraction, are stretched more than the strong sarcomeres. They're stretched to the point where they pop. And if you go back here, you can see that under the microscope there. Okay, we also see with MRIs that initially with a high, heavy resistance strain bout that sometimes the size of the muscle goes down once the swelling has dissipated. So now the weak sarcomeres are lost, and when you come back the next week, only the strong are there, so they don't get the damage. Then the next adaptation that happens, you add sarcomeres. So now all of them are strong, but the addition of sarcomeres is very important to prevent against damage. Why? Think of your typical stress-strain relationship, right? You have stress, you have a plastic point and a failure point, which is where we have the popping sarcomeres. If you have two sarcomeres and you go this length, these sarcomeres will be placed under more stress and be in danger of hitting the plastic or failure point. But if you have three, they're each stressed less. So that's why the addition of sarcomeres, which happens typically on lengthening contractions or longer range of motion, becomes very important to protect your muscles against damage. So you want to think about that when you're training. Make sure you're emphasizing a full range of motion if the subject or your client or yourself can do that. So how do we apply this? Now, oftentimes trainers and coaches go overboard on day one. You see it all the time, and what does that mean? We bring athletes in after the off season. It's like, oh, I got to make them mentally tough, so I'm gonna beat the living daylights out of them. But is that the right technique? Setting aside this mental aspiration that the coach has. There's a study by Brown, and basically what they did was they had 3 groups, and they were people who hadn't trained for a while. So you had one group where essentially they did 50 reps or 5 sets of 10. On day 1, you're looking at muscle damage. It goes up here. They did 3 sets of 10, muscle damage goes up again, and they did 1 set all out till failure for 10. The damage response was very low on the 1 set. But in the 3 and 5 set, damage was high, indicating that volume is important for damage. They brought them all back after recovering after 2 weeks, and they exposed them all to 3 sets of 10. Notice what happened. They all got the repeated bout effect. All of them. So what this says is that if you have an athlete coming off of the off season, you shouldn't beat the living daylights out of them the first week. You should bring them in, and during week 1, you should focus on technique and work your way up to 1 maximal set. The rest of the week should be focusing on skills and team cohesion. And then the following week, you can increase the volume and do your 2-a-days. This will prevent injury and losing your athletes. It will also increase team cohesion, and that will make the team stronger. Thank you.